Hola. Hola, 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 hola. Hola, 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 hola. Prueba, 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 prueba. Vale, perfecto.
Yes.
Um, eso es, perfecto. So, good morning to everybody and welcome to this new seminar of the Rocasolano Institute cycle of seminars, scientific seminars. As you know, today we have the pleasure to attend the seminar of Dr. Miguel Garvez, that everybody from the institute know him very well. He's a member of the institute. And let me introduce some items from his research career. Miguel Garvez obtained his degree in chemistry in the Universidad de Salamanca, and he made a master in molecular and cellular biology, and then he moved to Madrid, and he obtained his PhD degree in structural and molecular biology. And there, under the supervision, his supervisors were Dr. Alfredo Villasante from the Centro de Biología Molecular Severo Ochoa, and Dr. Carlos González from the Rocasolano Institute. His research has been almost always, correct me if I'm wrong, but on the structural and functional characterization of nucleic acids. After obtaining his PhD degree, he then moved to the Universidad de Salamanca. And then he came back in 2017 to the Instituto Rocasolano, and then he got Juan de la Cierva contract. Currently, just to summarize, he is a recipient of Marie Sklodowska-Curie Global Individual Fellowship, which entails a stay, 2 years stay in a country from abroad. In this case, he went to McGill University, to the Department of Chemistry in Montreal, Canada. Sorry, I forgot the name of your supervisor there. Masad Dama. Always focus his research on the chemistry of nucleic acids, and he's now in the Rocasolano Institute. So thank you very much, Miguel, and start with your talk.
Hi, good morning. Well, good morning to everyone here in the audience. Good morning to those that are following the seminar through streaming. Thank you, Josemi, for the introduction. Thank you, Josemi and Steph, for organizing this seminar series. It is for me an honor, it's a pleasure to be here, surrounded by friends and colleagues. And yes, so today my talk is going to be about nucleic acids. And let me please start by just the very beginning, by just the very basic concepts of nucleic acids chemistry. So I know that probably all this that I will tell you now, you already know because you have studied that in the high school, in the school even. But let me remind you some basic concepts because I think it will help you to follow the rest of the seminar. So nucleic acids are biopolymers. So they are polymers formed by monomers, which are nucleotides in this case. And the nucleotides have three main parts. So we can differentiate, a sugar moiety in a nucleotide, they're in green. Then there is a phosphate group, and finally there is a nitrogenous base. Okay, so the sugar used to be a deoxyribose ring of 5 members in the case of the DNA, and it is normally a ribose, and it is a ribose in the case of the RNA. The only difference between these two sugars is the presence of this hydroxyl group in the 2' position. The phosphate group is essential for linking consecutive nucleotides in the polymer. So it forms these phosphodiester bonds that then constitutes the backbone of a DNA or RNA strand. And finally, the nitrogenous bases that can be divided in purines or pyrimidines purines are adenine and guanines, and pyrimidines are cytosine and thymine, and in the case of the RNA is uracil instead of thymine. These nitrogenous bases, these chemical groups, are actually the alphabet, the letters of the alphabet in the genetic information, So as I say, the genetic information is written in the DNA. So what we have inside our nuclei, in all of our cells, is molecules, long polymers of DNA, comprised of around 3,500 millions of nucleotides. This is what we have in each of our cells. And the DNA is always in the nuclei, so it never leaves the nucleus. So for the genetic information to travel to the cytoplasm, we need to translate partially the genetic information from the DNA language to another language, which is slightly different, but pretty much very similar, which is the RNA language. So to do that, the cell uses a process called transcription. And the transcription consists in reading the DNA message, the DNA language, and translate it into the RNA language. So remember that in the new language we will have the OH group in the sugar, and then we will have uracils instead of thymines in the molecule. So the first result of the transcription is a pre-mRNA, which contains regions that will be read by the ribosome, which are the regions called exons. So these are regions that encode for a protein, and some other regions that will be deleted from the mature mRNA, which are the introns. Okay, so before this pre-mRNA can travel to the cytoplasm, it needs to be modified. So it has to have a poly-A tail, and it has to have some other modifications in the 5' end. When it is matured and the intron has been removed from the RNA, the RNA is ready to travel to the cytoplasm. And once in the cytoplasm, it is translated and a protein comes up. So let me stop here just for a minute to present you the first molecule that I will present today, that is based in a nucleic acid molecule and that can act as a therapy. In this case, I'm talking about the recent vaccines, the mRNA vaccines for COVID-19. So these vaccines that were designed by Pfizer and BioNTech and also from Moderna, they are actually just messenger RNA molecules that has been synthesized in vitro out of the cell, and they are encapsulated in these lipid coats, and they contain the information to synthesize the spike protein of the coronavirus. So basically, these kind of vaccines that are now a reality since a few months ago are injected into our bodies, and then this messenger RNA goes into the cell, and this process happens. So basically the translation of this messenger RNA gives rise to the spike protein, and then you know what happens. So the immune system reacts, generating these antibodies against the spike protein that protects us from a future infection. Well, it is important to know that these biopolymers, so nucleic acids, have a structure. So they are folded. And then the common feature of the folding of nucleic acids is that they are always helices. Okay, so they can be double helixes, triple helixes, quadruple helixes, etc. So let's begin by talking about the most common secondary structure of nucleic acids, which is the canonical double helixes, the A form and the B form of DNA and RNA. So this canonical nucleic acid structure has to fulfill three main characteristics, three main features. One of them is that they have to be anti-parallel. So they are double helixes in which the two strands run in opposite orientations. Okay, one of them goes from 5' to 3', and it associates with another one that runs in the opposite orientation. Then they have to be right-handed, so they have to grow to the right. We will see this a little bit more in detail later. And finally, they have to be stabilized by Watson-Crick base pairs. Okay, the Watson-Crick base pairs are these associations between bases. They are the guanine and cytosine, the GC base pair that happens between a guanine and a cytosine, and it is stabilized by these three. Hydrogen bonds, and then the adenine always is associated to a thymine through these two hydrogen bonds. Well, of course, in the RNA, instead of the thymine, we have the uracil. Okay, so here you have the two most common structures or helical structures of DNA, the B form that was discovered by Watson-Crick and Franklin in the 1953, so around 60 years ago. And here you have the most common helix formed by RNA, which is the A form. Okay, so if you want to differentiate them, one of the things that you can do is just you can see the structure from the top, and you will see that in the A form you see this channel, this white channel in the middle, that is not present in the B form. So I just said that this is the most common structure of DNA, and actually one of the most frequent strategies to combat disease or to treat some diseases, like for example cancer, is the disruption of this structure. So most of the chemotherapy strategies that has been developed are based in disrupting the essential structure of nucleic of this DNA. And basically, for example, some of the agents, some of the chemotherapeutic agents that are used now just consist in small molecules that interact with the guanines of each of the strands of the DNA. So they can create these linkages between guanines, and this disrupts completely the B form of DNA. So the result is a series of events like the guanine excision. I mean, some structural defects in the B form, in the B helix, that finally produces the apoptosis, in this case of the malignant cell. Maybe you're wondering if the same case is also valid for the RNA. So if the RNA is also a target for treating disease by small molecules. So actually there is just one example of a molecule that has been approved actually really recently, less than a year ago, that targets an RNA molecule. And this is the case of Evrysdi. Evrysdi was approved last August by the FDA to treat spinal muscular atrophy. Spinal muscular atrophy is a disease in which the patient is unable to synthesize or to express a gene called SMN1. SMN1 gives rise to a protein which is essential for the muscular maintenance. So in these patients, what happens is that they are not able to produce SMN1, but actually we have another gene which is a paralog, so encodes for exactly the same protein. The name of this gene is SMN2. It encodes for the same protein, but the gene has some differences. So actually there is one of the differences that appears here in the exon 7. It is the substitution of a thymine for a cytosine. So just this single difference provokes that the splicing machinery eliminates exon 7 from the mature mRNA. So the result is a truncated version of the gene, which gives rise to a protein which is not stable at all in the cytoplasm. So it degrades very, very quickly. So what does Evrysdi do? Evrysdi basically binds to the messenger RNA, to the pre-mRNA actually, exactly in the position in which the splicing machinery acts by removing the exon 7. So with the presence of the Evrysdi, the splicing machinery is not able anymore to remove exon 7. So the resulting mature mRNA contains exon 7, and the resulting protein is fully functional. So it's a full-length protein and fully functional. And in this way, the patient is able to compensate the lack of the SMN1 protein. So I have just said that there is just one example of a small molecule that has been approved to target an RNA molecule, but there is another alternative that has been proven to be a little bit more effective in terms of drug discovery, and this strategy is the so-called antisense strategy or antisense therapy. This therapy consists in using small oligonucleotides or short oligonucleotides with a sequence which is complementary to a certain region of a messenger RNA. So in this way we can bind this small oligonucleotide to a certain region of a messenger RNA. And when we introduce this kind of antisense oligonucleotide in a cell, we can trigger some different kind of processes. One of them is that the RNase enzyme, which is an exonuclease enzyme, recognizes this particular structure, which is a hybrid duplex, and recognize it as something that has to be eliminated. So it eliminates basically the mRNA part of this hybrid duplex. Another thing that we can do is designing an antisense oligonucleotide with a complementary strand of a region which is really important for splicing, for example. So if we design this, if we direct our antisense oligonucleotide towards this kind of sequences, then we can modulate also the splicing. So we can induce the inclusion or the exclusion of a given exon, for example. And another thing that we can do is just interfere in some of the processing of the mRNA, like for example polyadenylation process, just by designing an ASO, an antisense oligonucleotide against these regions implicated in polyadenylation of the mRNA. The ASO also can act in the cytoplasm of the cell. So imagine that we create here a hybrid duplex, and then this can be recognized by exonucleases like the RNase, and again producing the degradation of the mRNA, or even by another protein complex called RISC complex, which basically does a very similar mechanism. So it degrades the messenger RNA by leaving also untouched the antisense oligo. And another important thing that we can do is just direct our antisense oligo to a region which is crucial for translation. So in that way we can inhibit or we can block translation. For example, if we are interested in inhibiting translation of a mutant protein. So, as I said before, this strategy has been proven to be a bit more effective than just looking for small molecules. This is a list of the 10 oligonucleotides that has been approved as drugs, as treatments for different diseases. There are many others that are now in clinical trials, in different stages of clinical trials. As you can see, the first one was approved almost in 2000, in 1988, and now recently, in last year, there are several ones that has been approved for different conditions. So one of them, as you can see here in the square, is to treat spinal muscular atrophy. So basically, well, the name of this drug is nusinersen. And so we have an antisense oligonucleotide. Basically, the mechanism of this drug is just to target the region that I said before that was important for the splicing. So this drug creates here a hybrid duplex, and in this way, it is able to alter or to induce splicing modulation. So again. The effect, the result is the same. So we have a full-length messenger RNA, and we can obtain higher levels of the SMN2 protein. So basically, two strategies for the same disease, targeting RNA, both of them. One of them through small molecules, through a small molecule like FSD. The other one for by using antisense strategy. And well, any of them has its own pros and cons. probably the most important advantage of this kind of therapies of antisense oligonucleotides are their specificity. So they are very specific. We can drive the oligo to exactly to the place that we really want to be. We really want it to be. However, the main advantage for ABSD, however, is that is better distributed in the organism. So actually, this drug can be taken just by oral administration. So the patient can just take a liquid solution at home, and that's enough. But however, in the case of nusinersen, these compounds used to be quite unstable in blood, so they are not well distributed in the organism. So actually, it has to be administered by injection, by direct injection in the spinal column, which, as you can imagine, is quite painful and requires anesthesia and so on. So why that happens? Why these drugs are so unstable? Well, the reason is that these drugs are basically oligonucleotides, so they are recognized inside the cell by exonucleases and endonucleases, and they degrade this kind of material. So what it is needed to do to get a stable enough drug based on antisense oligonucleotides is including chemical modification in the structure. So here I'm showing you some of the chemical modifications that one can do in an antisense oligonucleotide. One of them, which is very common, is the one that you can do in the phosphate group. So actually, by just substituting this oxygen by a sulfur atom, the resulting oligonucleotide is much more stable. So it resists better the degradation by endo and exonucleases. And also, there is another important feature of this kind of oligos containing sulfur, or phosphorothioate, which is actually the name of this backbone, which is that they are more lipophilic. That means that they interact better with proteins, and this is important for a better distribution, a better internalization of the oligo. There are another kind of modifications that you can do, which are in the 2' position of the sugar. So in the 2' position of the sugar, you can include these kind of groups. For example, this is one of the most common ones, the 2' MOE, 2' methoxyethyl group. This is a bulky group that is very widely used in this kind of drugs. There is also the constrained ethyl, as you can see, it's like a bridge under the ribose ring. There is also the LNA locked nucleic acid, and also you can include a fluorine atom in the 2' position, like is the case of this modification, 2' fluoroarabinose. These kind of modifications provide resistance against nucleases. They also increase the binding affinity for the target, and also they reduce some pro-inflammatory effects that these drugs used to have. Well, we have been talking about the canonical structures of nucleic acids, the double helixes. We have seen the double-stranded DNA, the double-stranded RNA, and the hybrid duplex, which is so important for the antisense therapy. But I would like also to mention some other structures which are considered like the non-canonical structures of nucleic acids. So I said at the beginning that to be considered as a canonical helix, there was 3 features that have to be fulfilled. One of them is that the helix has to be antiparallel, and maybe you're wondering if there is a duplex which is not antiparallel, and the answer is yes. There is the parallel duplex, in which the 2 strands run in parallel, as you can see here, in parallel orientation. There is also some double helixes which are not right-handed, like is the case of the Z-DNA. The Z-DNA is a left-handed helix that appears in the DNA when there is a high concentration of salt in the environment. And I want to give you a trick just to remember if you have a left-handed or a right-handed helix. So basically, you can imagine that the helix is actually on a spiral staircase, and you can imagine that the steps of this staircase are the base pairs, and the backbone would be the stair rail. So basically, if you want to go up this staircase, you have to think which hand would you use to hold the stair rail. If you would use the right hand, that means you have a right-handed DNA or RNA, and vice versa. So if you have to use the left-handed to hold the stair rail, that means you have a left-handed helix. Finally, is there any structure, any double helix which is not stabilized by Watson-Crick base pairs? And also the answer is yes. So there is the Hoogsteen duplex. The Hoogsteen duplex is a duplex which is stabilized by Hoogsteen base pairs. So the Hoogsteen base pairs were described by Karst Hoogsteen, and they implied the formation of these base pairs in which the groups involved of the purines are not the same as the ones in the Watson-Crick. So this is the Watson-Crick phase of the purine of the guanine, while there is another two groups that can be involved in base pairing, which are these other two groups. This is what is called the Hoogsteen phase of the purines, adenine and guanine. So this kind of base pair is very important to form another very interesting structure of nucleic acid, which is the triple helix. So the triple helix is actually only possible because of the presence of a purine-rich sequence. So the purines have these two phases, the Watson-Crick phase and the Hoogsteen phase and the Watson-Crick phase. So if you have a strand full of purines that is interacting with a strand full of pyrimidines, so you have here a bDNA, a normal bDNA, you still have all the Hoogsteen phases available here to interact. So there is, if you put there a third sequence and as a third strand, you can hybridize it through Hoogsteen base pairs formation. And this is the way that a triplex is formed. And the triplex is also, as I said, very interesting structure of nucleic acids, because it is now well more or less established that this structure can be formed in vivo. Actually, it is widely demonstrated that by forming a triplex in certain positions of a gene, one can inhibit important cellular process. For example, if you form a triplex in a gene promoter by using an exogenous oligonucleotide, in this case we call them triplex forming oligonucleotide, then you can inhibit the transcription of that gene. So you can silence partially the transcription of the gene. And another strategy that is being using is the use of these kind of schemes in which you form a triplex here and you drive this donor duplex DNA towards a region of the DNA that you want to repair. So if there is a similarity between these two duplexes, it can happen an homologous recombination, and you can actually repair this part of the duplex. Also, it can be the same strategy can be used to introduce some mutations in the gene. I want also to mention another way to form a triplex, and it's just providing the cell with a Hoogsteen duplex. And this is a strategy that was designed and developed by Carlos Ciudad's group in Barcelona, in the University of Barcelona. What they use is actually the other half of the triplex. So they actually provide a duplex stabilized by Hoogsteen base pairs. And this Hoogsteen duplex, or Hoogsteen hairpin, actually is directed or is targeting a polypyrimidine region, so that here they form Watson-Crick base pairs while they already had the Hoogsteen pairs there. So the result is a triplex here that can be formed in a promoter or in another region, and you can, in this way, you can modulate transcription or modulate, for example, gene repair, etc. And let me just dedicate a few minutes to talk about these quadruple helixes. These are very interesting structures for us. It has been a topic of research in Carlos Gonzalez's group for a long time. And just let me tell you how do they look like. So basically, we can have quadruples. There's two types of quadruplex helixes. One of them is the G quadruplex, so the guanine quadruplex, and the other one is the intercalated motif, or the I motif. So the G quadruplex is basically formed by the stacking of this arrangement of 4 guanines. So this is called a G tetrad. So this kind of arrangement, stabilized by hydrogen bonds, as well as you can see here, if they stack on top of each other, they form a quadruplex. Okay, these structures used to be stabilized by the presence of monovalent cations, like is the case of potassium. So, and the other example is the other tetrahelical structure is the I motif, and the I motif is a very curious structure in which what we have is the intercalation of cytosine-cytosine base pairs. This structure, as I say, is based in the formation of this non-canonical base pair, which is the cytosine-cytosine base pair, and requires the prosonation of one of the cytosines in the pair. So this is why these structures used to be more stable at acidic pH. Well, it turns out that these structures have been detected inside the cells by the use of specific antibodies, and it's curious that they are particularly prone to be formed in certain regions of the genome, so particularly in some regions involved in regulatory roles. So for example, there are many Gs, many guanines in telomeres, many guanines in promoter sequences of some oncogenes as well, and also in ribosomal DNA, in some of the regulatory regions of messenger RNA. So these structures are prone to be formed in these regulatory regions. So that's why there is a whole field, a research field, which is dedicated to finding ligands which are specific for this kind of structures. This is just a representation, a bunch of them, of the ligands that have been designed and synthesized so far. Maybe you're wondering if there was some success in designing of this kind of small molecules targeting quadruplex. There was this molecule called quarfloxin. There was that reached phase 2 of clinical trials for treatment of a kind of tumor. And basically, the activity of this compound was it bound quite selectively G quadruplex appearing in the ribosomal DNA. So in this way, it was possible to inhibit the transcription of the ribosomal DNA. Okay, so finally, the I motif. Well, the I motif is a structure that was discovered more recently. So this field is less developed than the G quadruplex field. However, there is a great interest also in finding ligands, specific ligands for this kind of structure, because these structures has also been detected in important regions of the genome. And this is the end of the first part of the seminar, in which the main goal was just to convince you that acting on the structure of nucleic acids can be used to treat disease, and I hope I could do it. And now I'm going to try to explain you, or to show you how we have applied this philosophy in a project that is related to a neurodegenerative disease as amyotrophic lateral sclerosis. Amyotrophic lateral sclerosis, ALS, is a neurodegenerative disease that is caused by the extensive loss of motor neurons and glial cells in the brain and in the spinal cord. The result of this extensive loss of motor neurons causes horrible symptoms, like beginning with loss of motor control, stiffness and twitching of the muscles, and a progressive losing of motor control. So finally, this leads to a heart difficulty to swallow and to breathe, that used to cause the death of the patient within 2 or 5 years normally. So that was not the case of one of the most famous persons that had this disease. It was Stephen Hawking, that survived up to 55 years with ALS. That was a strange case. And then this person here, this guy here, is a baseball player that played in America in the last century. His name is Lou Gehrig, and his case, he was diagnosed with ALS also, and his case was so shocking for the American society that also the disease is also known as Lou Gehrig disease. There is no cure for ALS so far, so despite the great effort that has been done to find drugs or possible cures for the disease, there is no cure. There are just these two compounds that were approved by FDA, and that they have just a slight effect on the patient's survival time. This riluzole increases just around 3 months the life expectancy of the patient, and a double one that was approved a few years ago just slowed down the disease progression. And also it is not well known what is the exact cause of the disease. These are just a list of the genes that has been implicated to ALS. One of them is the one here in the square, called C9orf72. Is considered the most frequent genetic cause. So it appears in a high proportion of patients. This mutation appears in a high proportion of ALS patients. Let me explain you a little bit about this gene. So this gene is located in the chromosome 9. That's why it is called C9orf72. It's the chromosome 9 open reading frame 72. This is the, as you can see, it has 10 exons, and it turns out that there is a sequence in the first intron, which is a GC-rich sequence, GGGGCC and the complementary one, that appears repeated a number of times in every individual. So, and the normal number of repeats of these sequences is 2. So 2 repeats of this sequence, but it can go up to 20, 25 without problem. The thing is that in a high proportion of ALS patients, the number of repeats goes up to hundreds or thousands. So it increases exponentially. This DNA can be for sure transcribed into these RNAs, one of them containing a lot of G's, the other one containing a lot of C's. And these RNAs, the presence of these RNAs in the cell has different consequences. One of them is that these RNAs form aggregates inside the cell, and these aggregates are toxic for the cell. Another thing that happens is that these RNAs can sequester RNA binding proteins, so that these RNA binding proteins can no longer do their function. And finally, and very importantly, these RNAs can also be translated. They are translated into through a mechanism which is non-ATG dependent, so it's a very particular mechanism, and it gives rise to very particular proteins called dipeptide repeat proteins. So these proteins are basically repeats of 2 amino acids. So basically arginine and proline, for example, many times repeated in the protein. And these proteins have also toxic effects for the cell. So what we proposed was to use these RNAs containing these repeats as targets to find possible possible therapeutic approaches for ALS. So we have like 2 approaches. One of them is finding or trying to look for ligands that could bind these targets, these RNAs. In this way, we pretend to probably stop aggregation. Maybe we can stop TPR translation, or even we can occupy some binding sites of RNA binding proteins. And the other strategy, as you can imagine, is using antisense therapy, or at least try to design oligos that could be targeting these RNAs. In this way as well, the goals were the degradation of the toxic RNA or stop the DPR translation. The first thing that we did was to synthesize the RNAs, synthesize and purify RNAs containing the repeat expansion. Okay, so in this case we have prepared RNAs containing 16 repeats, and we have characterized the structure of these RNAs by Nmr spectroscopy. So you can see here the Nmr spectra of the G-rich RNA. It shows these signals between 10 and 12 ppm, which are characteristic of the G quadruplex formation, because these signals belong to these immunoprotons that appear in the tetrads. This is the circular dichroism spectra. which is also corresponding, which is a characteristic of a G quadruplex as well. So probably this RNA is forming some kind of G quadruplex structure. Then the other RNA, which contains also 16 repeats, we characterize it by NMR. You can see we can observe these signals between 12 and 14 ppm, and these signals are very characteristic of the Watson Crick base pairs formation. So probably this RNA is some kind of double helix, an A form of a double helix or a duplex. We wanted to explore a little bit more in detail the structure of these RNAs, so we ran these kind of native gels, and as you can see here, we have loaded the RNAs. We have loaded the RNAs in different conditions, and as you can see, there's in some cases there are some material that does not go into the gel. That means that there are aggregates that cannot enter through the pores of the gel. So we noticed that this happens especially when we heat the sample, when we heat the RNA and we let it cool down. So now we are trying to characterize these aggregates in vitro. We are using this. We are using AFM microscopy, and we obtain this kind of images. So this is an ongoing project that we want to explore more in detail. We want to use another kind of microscopies just to characterize better this kind of aggregation. So as I said before, our goal was to use, our strategy was to use or to find ligands against these targets, or to use antisense strategies. So today I have no time, and also we are still working in this part of the project, the one related to the RNA ligand screening. So today I will just talk about a little bit and very briefly about this part of the project, the antisense strategy. And this is the part of the project that I have been doing in Canada, in Professor Tama's laboratory, who is an expert in chemistry of nucleic acids. And basically what we have done is we have synthesized antisense oligonucleotides containing this kind of modifications. Okay, our goals is just to synthesize them and purify them. Also, we are interested in the structure. From the structural point of view, we have some questions that we want to answer. So, are these ASOs structured? What structures are forming? Are these ASOs able to form the hybrid duplex in vitro? And what is the stability of this hybrid duplex? These are open questions for us, and this is something that is normally not explored in the community of antisense therapy. Finally, we would like to test the most promising ASOs, antisense oligos, in cell models of the disease. I'm going to go very briefly through these results. This is the collection of antisense oligonucleotides that we have been synthesizing and preparing. As you can see, they contain different modifications and different schemes. So we combine DNA with 2'-fluoroarabinose. We combine different modifications just to see if they work good in vivo. Some of them are targeting the G-rich RNA, like the ones in the left. On the left, the other ones target the C-rich RNA. As I said before, we are interested in the structural part of so in the structure that these antisense oligos can form. And well, what we are doing is just we are analyzing the structure of the ASOs by Nmr Spectroscopy. And just here I can show you a couple of them. As you can see, some of these ASOs are quite well structured, so they form these structures. They give us these nice NMR spectra with signals between 12 and 14 ppm, characteristic, as I said before, of GC base pairs, and also it looks like they are quite stable structures. Some others, like for example this case, show these signals between 15 and 16. Let me see. Yes, 15 and 16 ppm, which are characteristic of these immunoprotons taking place in cytosine base pair. So probably some of the ASOs that we are dealing with are forming eye motif-like structures. Another question that we have, are these ASOs able to form the hybrid duplex in vitro, sorry. And to do that, to explore that, what we are doing is this kind of experiments. So they are native gels in which what we pretend is to identify or to see if this band is formed. So basically in this lane, what we have loaded is a mixed, a forced hybrid duplex that we have created just by heating both strands together in a tube. So we are sure that this is a hybrid duplex, and then we combine both the ASO and the target in different conditions, and we pretend to find if this band appears. And it looks like in most of the cases the hybrid duplex is forming in vitro. I don't know why this doesn't work. The other question was how stable was the hybrid duplex? To do that, to explore that, we are running these UV melting experiments. We obtain these kind of curves, and generally we obtain melting temperatures, very high melting temperatures. In this case, it was around 92, but there are some that are even bigger or higher. And finally, and this is the last slide, we wanted to test the most promising ASOs in models of the disease, and to do that we are collaborating with Dr. Keith Gagnon from the University of Illinois, of Southern Illinois, and what he does is that he has the cell model of the disease. So he has cells containing this repeat expansion in the C9orf72, and he is able to count the number of RNA aggregate foci present in the cell. So basically what he does is he counts the number of RNA foci in untreated cells, and then he counts the number of RNA foci after treating the cells with these ASOs. So basically some of the cases, the ones here in the square, these are the results for some of our ASOs. As you can see, in some cases there is a significant reduction in the number of RNA foci. Well, this is the last slide, but it is quite important. It is to say thank you to everybody that has been involved in this project and in other projects. I want to have a word for Alfredo. We miss him a lot. We are sure that he would have enjoyed a lot with all these projects, and I want to say that we really miss him every day here in the lab. Then a huge thanks to Carlos. I have no words to say thank you for his support and help during all these years. Thank you to the rest of the Nmr group of the IQFR. You're great. Thank you for your help and support as well. A huge thanks to Professor Dama in Canada. He's the perfect host, and also I consider him the perfect mentor as well. So thank you for being there and for treating me so well. To the rest of the Dama group, who are also fantastic. The collaborators involved in the project, and of course the funding from the Marie Curie Actions for giving me the opportunity of doing this project. I really encourage the young people to apply for the fellowship, which is a really good opportunity for any researcher. And that's it. Thank you so much for those that are here, for the people in YouTube, and I think that is time for question.
Okay, thank you. Thank you very much, Miguel, for this nice and revealing talk about nucleic acid. Thank you very much. So now it's time for question. Just to facilitate these questions and answers, we encourage you to make short and direct questions to the speaker. And those attending the talk via YouTube, please, you can use the chat to make these questions. Okay, thank you very much. So, yeah.
Sorry, the last part of the question I couldn't hear it. Yeah. Yes. Yeah, it codes for. Sorry, I don't have the. Sí. Hola. Oh yes. So Dr. Solis is asking that what is the protein that encodes the gene C9orf72? If I didn't understand wrong. Well, actually, the protein is called as the gene, so it's a protein called C9orf72, and it is not well known what is the function of the protein. It looks like it is not essential. The protein is not essential, but it has an important role in the motor neuron, because without the protein, the motor neuron is not happy at all. So. Yes, exactly. Yes, once you have the expansion, you don't have the functional protein anymore. That's true. Yes, there is a mixture of gain of function effects and loss of function effects that are involved in this mutation in particular. Hi, yes. So Doug, Dr. Lawrence, he is asking if given that the C9orf72 gene contains that amount of Gs of guanines once it happens this repeat expansion, if there is a problem, if that's supposed a problem to design drugs targeting this region, given that in telomeres there is a huge amount of guanines. And I don't know how to answer that. Actually, it is always tricky to design drugs against G-rich regions, because there are many places in the genome that you can find a lot of Gs, and also because you have to imagine that these regions are very prone to fall into G-quadruplex structures. So somehow, for example, if you're using the antisense therapy, you pretend that regions to unfold, so to form just a single strand RNA, and to form then the hybrid duplex. And this is not always that obvious and that direct, because you have to believe that the G-quadruplex structure is going to be, let's say, unfolded, and this region will prefer to form a hybrid duplex. So this is the main concern when you target these G-rich regions in the genome. But actually, when you design drugs against these G-rich sequences, for sure, it can go anywhere. So it is really difficult to design selective drugs in terms of a small molecule. I mean now selective drugs for RNA regions, and in particular regions, G-rich regions. Thank you. Yeah. That's very interesting. Dr. Oroth, he is asking if the G-quadruplex structure formation has an effect, for example, making difficult the transcription of some other processes inside cells, because he thinks that the G quadruplex structure is very stable, or at least more stable than a double helix. And it is not always like that, but yes, so it's true that normally G quadruplex are more stable than double helix, than the conventional double helix. So it is known that it is difficult to transcript in regions where there are a lot of Gs, in regions that can form G quadruplexes. So for the polymerase to go through these regions of the genome is quite difficult, and sometimes it is demonstrated that even in replication or in transcription, the polymerase is just stuck in that region. So they found the obstacle, they found the quadruplex, and they stop. Sometimes they go back a little bit and they then start to transcribe or to replicate again. But yes, they are actually obstacles in the genome. Yes. So there are also, it is known also that there is proteins, helicases, especially dedicated to unfold this kind of G quadruplex structure. So sometimes you require the action or the activity of a helicase or a topoisomerase or something, some kind of enzyme that allows you to unfold the G quadruplex, and then the transcription or the replication can happen. But yeah, that was a good point. Yes, I'm not sure. I know that in this case, in the experiments that, sorry, I didn't say the question to the audience in YouTube. So Dr. Mantegni, he's asking if there is a specific cell type in which you can observe the RNA aggregates or RNA foci. And I am not sure, but what I can tell you is that in the experiments that I showed here at the end of the talk, they're using induced pluripotent neuronal pluripotent cells, I think. So as far as I know, at least in neurons, they appear, and I guess in some other tissues, cerebral tissues, for sure they are present, but I cannot answer with more accuracy. Sorry. Thank you everyone. Thanks.
Lo podemos retrasar.